you know, I'm a little skeptical about the selective sweep hypothesis. Foster X effects are invoke partially recessive favourable mutations. So let's look at the possibility. I'm just coming to an end very soon, you may have it now. Effect of background selection on this. So the model I've used is very crude. We have a neutral site located in the middle of a Drosophila chromosome arm. Ignore the fact that the combination rates are not exactly uniform across the arm. And I'm also assuming that since we're dealing with the freely recombining part of the genome, we can keep the assumption that they're fairly strongly selected against. Now, the genome unfortunately is rather messy. We have non-synonymous sites, we have non-coding sites, which are under selection, and I've divided it up crudely into strongly selected non-synonymous sites, strongly selected non-coding sites, and weakly selected non-coding sites, according to information which I've culled from the literature. I'm not just making it up, not completely at least. And then I'm using estimates of the frequency distribution of mutational effects on fitness for these three classes of sites. So here's a cartoon. The model comes in several flavors. You can have introns or not have introns. I'm showing you a case where there's no introns. We've got intergenic sequence, which is divided into relatively short, strongly selected non-coding sequences, and longer, weakly selected non-coding sequence. And here we have the coding sequence in which the non-synonymous sites are subject to strong selection, with a distribution of selection coefficients that seems to correspond to the data. And these are the stats for the different assumptions. We also have to use map lengths, that's better known. Map length in females for Drosophila melanogaster is about 0.5 Morgans for an autosomal arm. Map length for the X is about 0.6 Morgans. So the population effective map lengths when we do the appropriate multiplication is about 0.25 for the autosome and 0.4 for the X. So I'm dividing the autosomal value by 2 and this one by multiplying it by 3/4. So in pseudo-obscura, the X has 2 arms, and there are 4 major autosomes with single arms. I'm just going to look at one of the 2 arms of the X. There's about twice as much recombination per base pair in pseudo as male. Mohammed's group have worked quite extensively on this. So the map length is about 1.2 for the autosome and 1.3 for the X in my model, and this gives us corresponding population effective map lengths, which we then plug in to our old friend, the background selection equation. And we need to sum up over the whole chromosome now and integrate over the distribution of mutational effects for each class of site and get the whole shebang for the predicted reduction in diversity for a neutral site sitting roughly in the middle of the chromosome and experiencing background selection for everything on either side. Now, it's a crude model. I don't pretend this is going to give you absolutely everything you want, but I think it's quite informative to consider it. There are various flavors of the model involving increasing levels of mathematical difficulty. So the boring one involves the standard Drosophila mapping function culled from the literature, and we just sum over all sites along the chromosome and integrate over the distribution of selection functions. Model 2 is the same, but we'd be a bit lazy. We just use a linear relationship between distance and recombination rate, otherwise the same as model 2. This is the crudest model. It uses an approximation which was introduced by Hudson and Kaplan, which involves assuming a linear map and that just integrates over a continuum of sites for the whole chromosome. This comes up with a remarkable result, which is derived from the basic equation, that the reduction in diversity is dependent only on the ratio of the total rate of mutations that are producing the background effects of background selection. So U is the total diploid mutation rate to mutations that are causing background selection effects, divided by the effective map length of the chromosome. It's actually a very remarkable, simple result. It does sometimes make you believe in God that things come out this simple. Model 4 involves an approximation which integrates over each cluster of sites along the whole chromosome for the same selection regime and then adding up the effects of each site. So it should be more accurate than this model because we're not just smearing everything along the whole chromosome, but we're actually considering each cluster of sites separately, but then we're integrating over the distribution of selection effect. Model 5 is the one which gave me the most trouble, It's quite complicated mathematically. This has now been recently published, so you can read all about it if you enjoy this kind of thing. It's an approximation which uses integration along the genome of the integral over each cluster of site, not a summation. And instead of using the entire distribution, I approximate by using only the first and second moments of the distribution of selection coefficients, which is a nice, juicy problem in maths, but otherwise not too interesting. Now, this is a very crowded slide. You don't need to bother with most of it. This is for melanogaster. These are the 5 models. Top part of each row is for the dominance coefficient of a half. That's totally additive. This is for partially recessive deleterious mutations. What I mean by the adjusted X over diversity ratio is 3/4 the value of the B values for X and A. In other words, that's what you would expect to see if you looked at nucleotide site diversity and there were no effect of sexual selection whatsoever. This is the bottom line, basically. The predicted XA over diversity values are about 0.9 for all of the models. This is the exact model, 0.935 with no dominance, 0.937 for recessive effects. There's only a trivial effect of dominance here. This is the lousy, quick and dirty approximation. That really agrees remarkably well with the more precise one. That really annoyed me, I must admit. And this one, where I went to a great deal of trouble, doesn't fit that well. This is for a case where there are no introns. Actually, if you put introns in, this model fits better than model 3, so it wasn't a total waste of time, but I won't show you those numbers. So for melanogaster, we're getting an X over diversity ratio, which is about 0.9. It's not too different from the data. Pseudo-obscura can do the same thing. The map lengths are much bigger, otherwise the model's the same. And we get a value which is about 0.8, about 80%. And that's not, again, significantly different from the data. So with the limited data we have available, these 2 different species with quite different map lengths seem to be coming out in approximately the right region of parameter space. So sum up here, pretty much out of time. Get pretty good agreement with the data on X over A diversity ratios for all 5 models. The exact model with a linear map gives almost identical results to one with a realistic mapping function, so you don't really have to bother about complexity of mapping functions here. Model 3 actually works rather well. It only involves the deleterious mutation rate and map lengths. That suggests that we don't have to worry too much about knowing all this stuff about distribution of selection coefficients, etc., for these broad brush comparisons. And of course, as you might expect from this result, the dominance coefficient doesn't play much of a role, provided it's not close to 0. What about humans? Nobody really cares about flies, I have to admit. We actually expect the opposite pattern. Because the X can't cross over with the Y in males, but the autosomes can cross over. There's a problem with mammals. There's this well-known higher mutation rate in the male germline, so we really have to normalize diversity values by interspecific divergence to get an estimate of X over A effective sizes or coalescent times, because of course the X has a different mutation rate than the autosomes because of this male mutation rate effect. But that's easy to take care of. Also, the mammalian genome is much more diffuse, large tracts of intergenic sequence, so it's not as easy to use model 3. But if we ignore that problem, then and use model 3, I'm just being lazy here, the ratio is about 0.73 instead of 0.75. It's much, much weaker effect than in Drosophila. How does that stack up against the data? Well, the data is a bit confusing as well. Overall, if there's a recent paper in Nature Genetics by Gotti-Patti et al., about 0.73 for African populations, which again are the ancestral ones. However, in Africans, the ratio actually comes out greater than 0.75 far away from genes. There's increasing evidence in humans that diversity increases as you get away from coding sequence. So if you're looking at non-coding sequence diversity, it seems to be seeing more hitchhiking effects near genes than elsewhere. Whether that's background selection or selective suites, we don't know for sure at the moment. But it certainly suggests other effects of hitchhiking on genetic diversity. So, we want to know whether this is all science fiction or not. The data I showed you is sort of reasonably consistent with the predictions of the model. Of course, there have been many instances in science where the data is consistent with the model, that the model is total bullshit. So this may be no exception. So we need better information, and I think that from what we've learned at this symposium, the modern techniques are really causing an explosion of this kind of information. And, of course, it's going to solve all these problems, and along with everything else. So I'm probably too old to see the final resolution of these issues in my lifetime, so it's up to you guys to go out and do it. And I'll finish here. This, of course, has involved help from an awful lot of people. I'm not going to read out the names, you can read them for yourself. And it also involves a certain amount of money. Much of this was kind of bootlegged off things which were, other things which I was supposed to be doing. Finally, last but not least, I'd really like to thank Mohammed Noor. He's organised a wonderful symposium. I think everybody's enjoyed it enormously. Thank you, Mohammed. I should explain, this is a takeaway around the corner from our flat in Edinburgh.